2025, the Nobel Prize in Physiology and Medicine was awarded for the discovery of T cells, or Treg, the important mechanism in immune tolerance, highlighting their critical role in preventing autoimmune diseases and advancing cancer research. However, despite their importance, accurately assessing of their functional state remains a challenging task for the modern immunology. Hi, my name is Arjana Rahmetova. I'm a student of Gymnasium 56, Almaty City, Kazakhstan, and in this video, I'll explore the machine learning application to address this problem. Today, cancer and immune-related diseases remain among the greatest challenges in global health care. According to the World Health Organization, more than 90 million new cancer cases are diagnosed worldwide every year, and nearly 10 million people die from the disease annually. In addition, autoimmune disorders affect approximately 5 to 8% of the global population, and their incidence continues to increase. In Kazakhstan, as many other countries, the burden of oncological and chronic immune diseases is growing, creating significant medical and socioeconomic challenges. Regulatory T cells, or also known as a Treg, are playing a crucial role in supporting homeostasis. They are preventing an organism from its own excessive immune responses. Their dysfunction can lead to different autoimmune diseases or cancer progression. Despite the fact that the study of the regulatory T cells began in the 1970s, many aspects of their biology remain unclear. In particular, the mechanism that determine why some Treg cells become highly suppressive while others lose their suppressive function are still not fully understood. Therefore, in this project, I aim to evaluate the function activity of the regulatory T cells using single-cell RNA sequencing data combined with a neural network database analysis. For this project, I have created a neural network that can classify all Treg cells for 3 categories: high, medium, and low. The methodology of that project, I used publicly available single-cell RNA sequencing dataset obtained from the Gene Expression Omnibus database. Then I used preprocessing and filtration according to the canonical Treg markers as FOXP3, CD25, and others to identify Treg cells. Then, program started process of classification. I applied dimensional reduction methods, including principal component analysis or PCA and UMAP to visualize the structure of a cellular population in a lower dimensional space. Next step was using a k-nearest neighborhood-based approach combined with a latent clustering algorithm to identify distinct cell subpopulation with the regulatory T cell group. You can see all results on your screen. It is clearly seen that T cell population is not homogeneous. It is divided into 15 distinct subpopulations, proving heterogeneity of Treg cells. These clusters represent cells with different functional characteristics, including varying levels of activation and suppressive activity. As a result, I get a neural network that has an 80% of accuracy. For an 80% with accuracy, it can predict the functional activity of Treg cells using single-cell RNA sequence. This classification of regulatory T cells is useful because it allows researchers to see the functional diversity within what was previously thought to be an uniform population. By identifying subpopulation with the different levels of activity, suppression, and exhaustion, we can better understand how the immune systems work in both health and disease. In practical terms, identify a clinically relevant cell subpopulation that might serve as biomarkers, improve the design of personalized immunotherapies, help predict disease progression by understanding which Treg subpopulation dominate in specific conditions, support border research in precision medicine by showing how single-cell data combined with the computational methods can uncover patterns not visible with traditional analysis. Thanks for watching.